To Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. I am an associate professor of anesthesiology at the Medical College of Georgia. And today we are going to be talking about two interesting topics that are interrelated, nutrition and epigenetic programming. So we have the privilege of having with us Dr. Carsten Carlberg. He's a professor of Nutrigenomics at the Institute of Animal Reproduction and Food Research of the Polish Academy of Sciences. He's an expert in the topic, and it's a privilege to have him here today. Welcome, Dr. Carlberg.
Yeah, welcome.
Thank you so much. So we are going to base our discussion on a paper that was published in Current Opinion. This paper is titled Nutrition and Epigenetic Programming. And I want to start by asking Dr. Carlberg, for a lay person like myself, what is a good definition for epigenetics? Something that make us understand the basic ground to start our conversation.
Yeah. So if you take the term genetics, which in simple way is the DNA that we have in each of our cells and the nucleus of each of our cells, the preterm epi comes from Greek and means above. So something above our genome is epigenetics. So if we're talking later on about epigenetic changes, then it means it is not changing our genome, but it's changing something above our genome, which you will see in a moment is more the packaging of our genome. Because if you take our complete genome, which is 3 billion bases, it has to fit into a very small nucleus of, let's say, 10 micrometer. And micro is 1 divided by a million meters, so tiny. You can't see it by naked eye. And the DNA, our genome, is about 1 meter in length. So imagine if you have just simply the task to condense it, you already need something above the genome. But this is just structural aspect, and there is far more in there. It's not just the point of getting something 1 meter into a very tiny dimension, but it's a part of it.
I see. So the condensation of that genetic material is what makes epigenetics a relevant aspect, and understanding that not all the genes that we have, or better yet, not all the genetic information that we have is all the time coding for something, right? So there are parts of the genome that are not really coding, but are equally important. So my next question is, you mentioned that in that packaging of the DNA, the 3D structuring of the DNA, as well as the proteins that keep them together, named the histones, and the DNA itself, are important in the maintenance of this epigenetic equilibrium, right?
Yeah. So to bring up one term is chromatin. And chromatin is the name for the complex of our genomic DNA with proteins. You already named them, histone protein. You know? So the protein DNA complex is called chromatin, and this chromatin is what we basically have in the nucleus. I guess most of you, of the people who listen to this, have seen a chromosome in a textbook or maybe even under the microscope. And a chromosome is basically made of chromatin. A chromosome is not just DNA, it's a protein DNA complex, which in fact is very dense. But the fact that during cell division, chromosomes condensate and visible to us under the microscope is an epigenetic effect. Because if you're looking on a normal cell, you will not see any chromosomes. These are only visible in a very short time of the cell division, about an hour or so. And also this is important. If you just take it as an example, during cell division, our genome has divided to new daughter cells. In fact, the genome has first replicated, so we have double copy of our genome and our cells in a certain phase after the so-called S phase of the cell cycle. And then the task is to condensate both copies accurately so that they are easy to separate. And then you can put them really one by one to the new daughter cells. So this needs this is an epigenetic effect. But while the chromosomes are so condensated, there is basically nothing happening concerning gene regulation. It's like you're moving from one apartment to the other. In this day, where all your belongings is in the truck, there is no cooking. Kitchen and all the stuff is packed. So that day is no cooking. In the same way, in the moment when there is the metaphases of the cell cycle, there is no gene regulation. And this has to be short because the cell depends on gene regulation. So this is kind of overboarding epigenetics, very short time, and shutting off all genes for about an hour or so. But they have to be reopened, otherwise the cells can't survive.
So this epigenetic regulation, in this particular case of cell division, is kind of suppressing the regulation over the genes. Do we have an example in which the opposite happens, in which this regulation is actually enhanced?
Look, take an embryonic stem cell, which is a cell which is completely undecided. It can get any other cell of our body. So I'm talking the very early phase of embryogenesis. So in this time, the epigenome is maximal open. And epigenome is the genome-wide view on epigenetics. So these cells are maximal flexible, and then they have to take decisions, whether they want to get a neuron, a kidney cell, name any other of the 400 cell types of our body. And then part of the chromatin gets condensated in order to prevent that, for example, in a brain, there are the genes of the kidney active and we are producing urine in the brain. Would make no sense at all. And the other way around, of course, the kidney cell knows exactly what to do as a kidney cell. So that means from the whole set of the genes that we have, all the different functions that our body can do, and the embryonic stem cell, most of it's open. And then when the cells are differentiating, the respective subsets, the lineage-specific genes that you need for kidney or for neuron or any other cell type, they stay open, but all other genes are shut off by epigenetic events. They are then packed very densely in chromatin and are not accessible, a bit comparable to what we just spoke about in the metaphase of mitosis.
I see. And what are the most important biochemical processes that actually trigger those effects of putting the genes on and off during this epigenetic regulation? What is actually happening at that level?
So there are 3 different levels of the epigenome. The first level, which maybe most people, in particular with medical background, know, is DNA methylation. So that's cytosine. Cytosines are one of the 4 bases forming our DNA. These can be methylated. And when this happens, then we have an epigenetic change. In fact, most of the cytosines of our genome are methylated. So our genome is largely methylated and just is kept unmethylated in particular regions where regulation happens, like promoter regions, regulatory regions of genes. So DNA methylation is level number 1. Level number 2 are these histone proteins you mentioned. So the other component of the chromatin. These histone proteins form complexes, altogether 8 together form a so-called nucleosome core. And these histone proteins, they have a lot of lysine amino acids. And these amino acids can be modified post-translationally. For example, it can be a methyl group, an acetyl group, and also a couple of other groups be added there. So there's a huge amount of post-translational modification. And there is a code called histone code, which is kind of an alphabet for this different type of modification. So this is for one way how we can store information in the epigenome by changing the protein component in the one or the other way of methylation, acetylation, and similar. And the third value, the third level, is the three-dimensional organization. So, for example, if the DNA forms loops, and if there's a large loop, and so the three-dimensional arrangement within the nucleus is an important aspect of the epigenome. These three levels come together, interchange each other, so you can't really separate them, and altogether they're able to store information. It's a tremendous amount of storage capacity that every cell has in form of its epigenome.
So when those processes happen, the genes are regulated. So for some, I assume for some aspects, a higher degree of methylation is needed compared to other functions, right? It's not like the gene function depends on either methylation or the opposite, right? Both happen at the same time. Am I right?
Yeah, I mean, you have to look where in the genome you're looking. I just said at regulatory regions, they are mostly unregulated, unmethylated. Yeah, while for example, the non-coding regions of our genome, we have about 50 or 54% of our genome, which is more or less junk DNA, and this kind of junk DNA is largely methylated. And there, basically, the methylation prevents that this junk DNA is somehow accessible.
I see. So and it's obviously important to keep it that way to maintain health, depending on the situation, of course. But
for example,
go ahead, please.
Sorry to interrupt, but it's an important aspect that you just mentioned. It is not staying constant. For example, during aging, the methylation level and our cells changing. If people get cancer, the methylation level are changing. So there is during onset of disease or during progression of disease, as well as during normal aging, there are changes in this methylation. And they have, of course, an impact on the progression. It's information, and sometimes it's kind of bad information, sometimes it's good information. And the good thing is we can't change the genome, but we can change the epigenome to a large extent by our lifestyle. And this is what I guess it's a core thing we're talking about, is epigenetic programming. So if we change our lifestyle, we can change our epigenome, and that can, if we are lucky, not only if we're lucky, if we do it correctly, counteract certain diseases. So, for example, if I may be unlucky and get a cancer diagnosis today or tomorrow, first I will tell me, oh, I have done something wrong. It didn't happen for nothing. It happened because in my life, I seem to have done mistakes. These mistakes haven't been corrected by my immune system, and now I have to live with it. So maybe my immune system was, for the one or the other moment, not strong enough. So I have to retrain my immune system, and that goes primarily by inducing epigenetic changes to my immune system. So I would have to change my lifestyle. That means cancer diagnosis, I may have not done enough steps per day. I may have had too much stress in my life. I may have eaten the wrong things. So I need to do drastic changes to the epigenome in order to help my body, in particular my immune system, to counteract the disease, cancer or whatever other disease I may get.
And this is actually very exciting because it's like a new approach to therapeutics, if you will. It's the ability that all the information that we have in that epigenome can start intervening in your favor, and how you can implement some changes in the lifestyle and achieve an effect based on that epigenetic regulation. That is exciting. And actually, if you think about it, the genome and the epigenome, for that matter, are not in isolation. They are embedded in a milieu of metabolic pathways, if you will, and that leads me to my next question. So, where is the link between the metabolic intermediaries and the epigenome?
So these different modification of the histone proteins we just mentioned are done by enzymes, histone modifying enzymes. And there are enzymes who add the group, let's say an acetyl group, and other enzymes do the opposite, they remove an acetyl group. And this adding or removing is changing the chromatin. If the chromatin is largely acetylated, it's open. When the acetyl groups are gone, the chromatin is more condensed. So these enzymes themselves, they are like all other enzymes in biochemistry, are regulated by cofactors. For example, for the enzymes who are removing the acetyl groups, these are the histone deacetylases, or in short HDACs, they can be modulated by a level of the acetyl-CoA that we have in our body. This is something we get from normal nutrition. Sugar as well as fat are kind of digested in biochemistry for acetyl groups, and then together with a coenzyme, there's this acetyl-CoA. So this is the substrate for this histone-modifying enzyme. And there's certain form of this histone-modifying enzyme, a special form of the histone deacetylase called sirtuins, which are modulated by NAD8. And NAD8, NAD plus, which as is coming together with NAD8, is a transporter of electrons in our body. So if we are fasting, we have a lot of this NAD plus. If we are eating, if we have just eaten a meal, we have a lot of NAD8. Both come together. It's just saying fasting versus eating. So the fasting version of this cofactor, as it is modulating the activity of this histone deacetylase. And in this way, by simply fasting or eating, we are influencing our epigenome.
Yeah, it's interesting. So there is a direct effect of something that you can do. So to a certain extent, you have some control over the whole process, right?
You should have, yes.
You should have
your
exactly.
life, and
yeah, that's absolutely true. And there's something that I want to discuss now, and has to do with a concept that you mentioned in your paper about epigenetic memory. So it's not that you have like a one-time change in the epigenome and that's it, but the epigenome can learn. Is that a good way to see it?
Yeah, I mean, there is storage capacity in the epigenome. We have 30 million nucleosome complexes in each of our cells, each of them formed by 8 histone proteins, and the histone proteins can be modified at about 130 different positions. So if each modification is just 1 bit, and if you just multiply it, you have in each cell many gigabytes of storage capacity. So if you know how our body is made of 10 to the power of 13 cells, this is a number that most of us have basically no idea. So a huge amount, and each single cell can already store a gigabyte. So then you could just imagine the storage capacity of our cells, which is far more than a big server. So if these cells kind of shoot a movie, if you want to take this analogy, of our lives. I mean, sometimes you see boring thing like what we're eating, how well we are sleeping, what we are doing, if how much physical activity we have. So definitely the muscle record more about the physical activity, and maybe the liver more about what we're eating. And it's a kind of memory which you can compare to if you think about memory of your childhood, which may be 50 years or so ago. So this information, 50 years old, how was it kept in your brain? There must be a change to a molecule which is staying 50 years with you. Otherwise, it would not be accessible. So it can't be a protein. It can't be a lipid. It can't be a carbohydrate. It is something related with the DNA. And it's not the mutation. You will not have to memory a lot of mutations in your neurons, in the genome of your neurons. It's changes of the epigenome. So if you take this analogy that you can restore memories from your early childhood, let's say from 5 years, 6 years old, or whenever it really kicks in that you remember things, in the same way, your liver, your muscle, using the same principle of memorizing in the epigenome information, can basically remember what you had done as physical activity, and if you have eaten well throughout your life. It's also kind of a repetition. I mean, like whatever, certain cameras we have nowadays everywhere at shops or whatever, where they are just recording if something has happened, somebody has stolen something, whatever they are recording. But they normally overwrite it every day or every week or whatever to have not constantly keeping the information. But certain things which are repeating, of course, kind of getting enhanced. You do eat every day the same thing, a certain amount of steps, 10,000 steps. You eat good, healthy things every morning or every day, every time you're eating. This is kind of confirming the information. If you then once escape to McDonald's or similar fast food, it's probably not harming. But if you visit McDonald's and similar every day, it makes a constant memory of it. So the repetition does it, and that is then keeping a stronger and stronger memory of what what lifestyle you have.
And that is at the individual level. Can it be transgenerational as well?
Yes, of course, only until you have children. So after you had your children, it's just your personal business. But of course, before you have children, whatever you do can also affect the epigenome of your gametes, and then there is a chance to give it to next generation. Although during embryogenesis, there is a large demethylation, for example, of the genome, but it's not 100%. There certain information is kept, and this kept information is epigenetic programming of your parents until the moment of conception. So until then, yes, your parents could have collected information in their epigenome, which to a certain extent is in your epigenome.
Wow, that is exciting, and I want to go back a little bit to the nutritional aspect of the epigenome. I want to learn more about the effect of micronutrients on this epigenetic regulation. Can you tell us a little bit about it?
In my favorite micronutrient is vitamin D, which is a compound that we produce in our skin when we expose our skin to UV at an intensity that is high enough. That means in Northern Hemisphere at the moment, maybe in Georgia, but at least not in Europe, there is at the moment a chance, even if it's sunny outside, to make vitamin D. So the more north you live on the planet in winter, the less chance you have to make vitamin D. So you have to take it up by a diet, which is not that rich in vitamin D if you're not eating fatty fish a lot, or by daily supplementation. And that is basically recommended in Nordic countries. I was and still am affiliated with the University of Eastern Finland. And in Finland, one of the most northern countries on the planet, you can imagine in winter, everyone has to supplement vitamin D because for 5 months, there's no chance that there will be vitamin D made in the skin. So this vitamin D, when it's a bit more modified to a hydroxy group added to this vitamin D, then it's an hormone which binds with a very high affinity to a transcription factor called the vitamin D receptor. And this vitamin D receptor is one of 1,600 transcription factors that are encoded by our genome. And this vitamin D, or the biological active form, this 1,25-dihydroxyvitamin D, is modulating the activity of this transcription factor. The vitamin D receptor belongs to the family of transcription factor to which, for example, also the estrogen receptor or the glucocorticoid receptor is belonging. So if you know the impact of what estrogen has, in particular in females, or cortisol on stress, so the general impact in disease as well as in health, it's from the mechanism a very similar way. But the origin here is not an hormone that we are producing ourselves, but it's something that initially is produced in our skin and taken up as a micronutrient. You are not eating cortisol, you're not eating estrogens, basically. Of course, you can take up pills containing estrogens and so on. And of course, you can take cortisol or dexamethasone as a drug. But you are having with vitamin D and micronutrients something that you basically need every day, and that is regulating gene expression.
So the vitamin D is has the ability of let's say, keep the epigenomic regulation in check, if you will, right? So it's something that you need to have in order for these processes to be regulated, right?
Yes. So when I said that vitamin D is regulating a transcription factor, it automatically implements that this affects also the epigenome. So it is involved in opening, for example, the chromatin and changing the packaging of the DNA, changing the histomodification pattern of our DNA at the sites where this transcription factor is binding.
Yeah, and this is a very nice way that you have to explain it, and definitely you make it very clear for people who are not experts as myself. And I want to probably wrap up our conversation by asking you, based on your paper, what is the takeaway that you want people to remember for some, because these are new concepts, right, for the majority of people. What is the message that you want to leave?
The simplest message is you have things in your own hand. And if I have a rather simple rule, we haven't spoken yet about it, but 80% of the risk of getting a disease or the impact for aging is coming from our epigenome or on our environment, and only about 20% from our genes. So this is, of course, you can't take it for every exactly 80-20, but so there are a few diseases who are clearly genetically driven. Think about the one or the other monogenetic disease. But I'm talking more about common diseases like type 2 diabetes, atherosclerosis, but also Alzheimer's disease, cancer, so that the larger part of the disease, the reason for that disease comes from the environment and the epigenome, and not too much what we are born with. And that means what we are doing throughout our life is important for our risk for the one or the other disease. And when I said that I may get a cancer or whatever other disease diagnosis, then if I take this concept, I have to say, I have done, because at 80% it was my fault, Yeah, I may have done something wrong in my life. Okay, I hope it will never happen. Yeah, and therefore, I mean, at least I can say in 35 years of working, I have never been any single day sick leave. Yeah, so it seems to work. Yeah, and
Absolutely.
I hope it works also for the next 60 years or so. Yeah. But anyway, it means that we are aware of our responsibility. That if we have a healthy lifestyle, if we're aware of it, that we get not ill by bad luck, we get ill because we did a number of mistakes. Yeah? So we can't blame our parents that they gave us bad genes, at least only to a small extent. We more have to blame ourselves that we did certain things wrong. And unfortunately, there are a lot of things that we can do wrong. We don't keep our body mass index under control. We may smoke. We may eat unhealthy. We allow too much stress in our life. All these things that everyone basically know what we should not do, but many people say, okay, let's do it tomorrow. It's another day. Yeah. So this is something you really have to be aware of. It's our own responsibility. It's our life. Yeah. A doctor can give us here and there some help, but at the end, they don't can't give us wonders. So it's our own responsibility. Yeah.
Yeah, absolutely. The impact of the lifestyle on the disease is very clear, as you mentioned, 80% approximately, for some diseases even more. And the good news is that it's never late to start changing your lifestyle, right?
Exactly.
And
Exactly.
start taking advantage of this epigenomic programming, right?
Yes. Yes.
Excellent. This was really a pleasure talking to you, Dr. Carlberg, and I appreciate you sharing your wisdom with us. And I am completely sure that our audience is going to enjoy all this conversation. Thank you very much, and
You're
we will
welcome.
follow up on your research, and I'm sure we will our roads will cross again in the future. Thank you so
Yeah,
much, and
you're welcome.
for our audience, you will find the link to the paper in the description of the video. See you in the next
Okay.
episode.